Myself as an LLB tutor and an admin. Before going further, let me say about myself. I did my masters in Madurai Kamaraj University and currently pursuing PhD in VIT Vellore. Okay, in this video, I'm very proud to launch our new teaching source, a teaching platform called Scientifically.
On a daily basis, we are planned to put videos. So these videos are welcome as short and selfie videos from the viewers. If you ask me what is bioinformatics, I'll refer to this example of 4 blind persons who are touching the elephant. They have never seen elephant in their life. Blind person touches the tail and felt it is a rope. Another guy who touched the tummy of the elephant felt it is a wall. Similarly, another person felt it is a tree. Someone felt like a snake, spear, and one guy who is touching the ears is saying it's a fan. The same way, bioinformatics can't be controlled or can't be run in a single circle, right? As if the blind persons are feeling in each and every part of bioinformatics, we are feeling the domain in different way. Bioinformatics are of different domains like algorithms, methods, pattern recognition, genomics, proteomics, and a number of things. So it refers a deep understanding of fundamentals. So for example, any beginner of bioinformatics might have conducted this experiment called multiple sequence alignment. And we might have stuck with the symbols like star, colon, semicolon. So this requires knowledge of amino acids. So amino acids are of 20 amino acids are there, protein forming amino acids. Each one will have 1 letter code. So to know the 1 letter code is important. And their functional groups, in which functional group they are aligned, like non-polar, aliphatic, aromatic, or polar, charged or uncharged. So these things will be confusing you. You might have seen this picture in Leninger book, right? So let me make you simple how to understand that. We are going to use mnemonics. In our school days, we might have known about BibGR, right? We can say violet, indigo, blue, green, yellow, orange, red, even during our sleep time. So that much memory created because of mnemonics technique. Same mnemonics technique we are going to apply for this 20 amino acids. So we all know it have been classified into 4 things: non-polar, aliphatic, aromatic, uncharged, and charged. So what I'm going to say is a monk called Gavlib is wearing a fully yellow vesti. He's a scheduled tribe monk. After his death, that monk have been buried in a round hole. Okay, we are digging the round hole and burying. So it will be easy for you, right? If you look at non-polar aliphatic, that is called Gavlib. So Gavlib stands for glycine, alanine, valine, leucine, isoleucine, proline. I'm saying about the 1 letter code. Similarly, if you look at non-polar aromatic, fully yellow vesti. So FYW stands for phenylalanine, tyrosine, tryptophan. F for phenylalanine, Y for tyrosine, and W for tryptophan. Same way, you can say for polar uncharged, like STC, serine, threonine, cysteine, methionine, asparagine, and glutamine. N for asparagine and Q for glutamine. Fine. Way the polar charged are DEC RH, digging the round hole. RH stands for round hole, whereas arginine and histidine. Whereas DE, aspartic acid and glutamic acid. So these 2 acids are acids, right? So they are negatively charged. Whereas KRH, K lysine, R arginine, H histidine is basic amino acids. So this classification will help you to understand, my dear friends. Mnemonics is one of the powerful technique for understanding any biological term. Now let us come back to this analysis again. See, the star have been given indicating it is identical. Look at the 4 sequences, so 5 sequences. P means stands for proline everywhere. So to refer identical, they are given star symbol. But whereas if you look at colon, where they are given S for serine, K for lysine, E for aspartic acid, and so on. So they belong to a partially conserved because they belong to the same functional group, right? Serine, lysine, all are polar. So they are partially right. So they are given under colon, semicolon. But if you look at colon, it is similar, where aspartic acid, glutamic acid, see D and E are what? They belong to negatively charged amino acids. So functional groups are even more same compared to the polar, charged and uncharged. You are getting the point, I hope so. So there are 3 symbols: star, colon, and semicolon and colon. Whereas star refers to identity and colon refers to similarity and semicolon refers to half similar. So these things, remembering 1 letter code will help us to understand again You all might be familiar with this picture, central dogma of life, right? Where DNA is converting into mRNA, messenger RNA, through transcription. That mRNA from nucleus is transmitted towards cytoplasm, okay, transported towards cytoplasm, where it meets endoplasmic reticulum, and endoplasmic reticulum have been met with tRNA, okay, in endoplasmic reticulum, particularly ribosomes, where it will be recruiting tRNA. The tRNA consists of amino acids. So how the transfer occurs, means a translation occurs. The tRNA will have anticodon, which will be checked with codon, and finally amino acid is synthesized as a peptide. So my dear friends, we all know this central dogma of life, but for a beginner to understand, it will be confusing, right? So I come with another technique called analogy. So in our childhood, we might have compared things, so for an easy understanding. Your school teacher might ask, what's the shape of kidney? You may say yes, it's a bean shape, because we have never seen kidney, but bean is visible for us. In the same note, I am going to compare Central Dogma of Life with Iron movie. Hope you might have seen Surya movie, right? Iron, South Indian Tamil movie. So that movie sequence I am going to compare with this DNA to RNA, RNA to protein. That movie, Prabhu, the character, will ask Surya to go and collect the diamond. So it's a kind of a diamond mafia, where what I refer here is India's nucleus and Prabhu's DNA and Surya's mRNA. Because Prabhu can't go directly, right? Because of his bulk nature. So DNA can't move out of nucleus because of its double helical nature, whereas mRNA can easily move. So I refer here as mRNA Surya. Surya carries half tone rupee note, right? So it refers to codon, which is given by Prabhu to go and show to the terrorist to get the diamond. So codon is refers note. Okay. With a half note, Surya reaches Africa. So Africa here referred as endoplasmic reticulum, and Congo is ribosome. That particularly rough endoplasmic reticulum, and Congo is ribosome present over there. Similarly, the terrorist will have refer as tRNA, and the note which had by terrorist is called as anticodon, another half note. The terrorist checks the codon and anticodon. Say check the two notes same. The same way, tRNA checks the anticodon with the codon present in the mRNA. So that's what I referred here. If both the notes match, the diamond will be handed over to Surya. The same way, if codon matches with anticodon, amino acid will release, right, from the top of tRNA. And one by one, the tRNA synthesize a peptide using amino acid tRNA synthetase. Fine. So that's what we referred with analogy of central dogma of life with iron movie, my dear friends. Hope you enjoyed.